Good afternoon, everybody.
Now we are live on YouTube also, sir.
So shall I start?
That helps me. Just I'll give you a few words of introduction and then hand over to you. Good afternoon, everybody. Welcome to this afternoon session. Today we have with us Dr. Mitali Mukherjee, an outstanding scientist working at the IGIB, Institute of Genomics and Integrative Biology in Delhi. A Shanti Swarup Bhatnagar Awardee, a prestigious award in Indian science. She is a PhD from Indian Institute of Science, works on human genome variation, particularly Indian human genome variations, and also an active member of the Ayur Genomics. So she is the best person to talk about human genome, and she will be dealing with primarily on the human genome, and also maybe touch upon some of our works in the process of explaining this one. Now I hand over to Dr. Mukherjee. You have free time. In the sense, all our past this one have been.
Okay. Thank you so much.
Not rush. Please go ahead.
Thank you so much, sir. Am I audible?
Yeah, we'll take the questions at the end. I'll be noting down all the questions.
Sure, sure. Thank you so much, sir. It's a pleasure to be giving a lecture in this forum. I've been also associated with IRI. I did my master's from IRI and I was Dr. Srinivasan's first student. So that way, Dr. Chopra was our head of the department at that time. So it's an honor to speak now. And I'll just share my screen. Is this visible? Is this visible?
Yes, yes, fine.
Yes, yes.
Okay. So, so good afternoon, everybody. I come from this institute called Institute of Genomics and Integrative Biology. We have two campuses. This is the Mathura Road campus, and after the lecture, after the lockdown, if you people are interested in visiting our institute and having more hands-on idea about genomics, you're welcome to our institute. So, the talk title goes as applications of genomics in human, and I would try to make it a popular talk so that you actually get curious about what genomics is all about. Though I've been told that I have to deal with certain sections of the book, I will deal with that in the course of this popular lecture. The idea is more because genomics is a very dynamic field, and all your students who are working in this area also might need to go back and have their curiosity satisfied with so much references in or so much information all around. So I'll just give a flavor of this area, and whatever are written in textbooks, every 2 years more and more information gets added to it. So I will deal with what is given in the book and what it has to offer, just by way of your interest. So I'll try to start with posing a few questions. That is, there are lots of questions kids ask, right? What makes us human? Why do we have such shades of skin color? Are apes really our ancestors? How do you know that? Do we all hear historically that we come from Africa? Do we really come from Africa? How can we question that, right? Or when kids are blamed for laziness, aggression, getting up late in the morning, is that a habit or is there something more to it? We have microbes all around us. Do they assume importance? Or also such kind of questions as if, when did we start wearing clothes? That's a question we really do not know whether we have the answers from a great deal of evidence, because fibers are things which have not survived in during all the archaeological excavations, when it is done, fibers are never found because it would not kind of stay that length of time. The other things are like, there are so many animals in the forest. Do we have a count of them? Are there ways to count them? Uh, uh, then there are other things. For example, why do we want to study if bats have infection? Now, the coronavirus time, bats have become important, but the question is that if bats die, does it do anything to our ecosystem? Other kinds of questions where watermelon, why there is so much of water, meaning what makes it accumulate so much of water, or in the honeybees, why if there are the same bee, why one becomes queen, one becomes drone, one becomes worker or forager. So why is there a community structure in the beehives? What happens? How do we survive extreme environments? Or can we actually know how animals go extinct? So can we learn that today and sort of have a conservation system built around that, so that we are able to um, save animals beyond all the climate change and all the other things? Are there other cues which we can get to know which animal are on the brink of extinction? Or the other things is, I like the coronavirus time, right? Everybody is looking for cure for corona and they want to solve it fast. Is there a way by which we could accelerate these findings? How do we find cure for genetic disorders? Not many people want to work on rare genetic disorder because they impact 1 in 10,000 people. That doesn't make business sense, right? But families who suffer with rare genetic disorder, to them that matters quite a lot, right? Or the other thing is, now we are all on the net. We are all heavily using a lot of digital media, and we will surely run out of space after some time. So how do we take care of compacting information so that we can actually have more space available to us? Or the last question is that we have this question always asked, that there is this ancient system of medicine like Ayurveda. Is it mythology or is it science? It is practiced today, but the people are not aware whether that was just a story which was written or there is something scientific behind it. So, how do you query all these, right? So, one of the ways by which you can query many of these questions can be by this area of genomics. So, I would tell you some of the stories which will make you think that there is so much more which can be done with genomics, and many of the answers we can get. Okay. So, normally this is an interactive thing. I will just leave a little pause and I will say that all these letters which are written over here, right, sentences, I'm sure all of you in the audience, none of you might have been able to read all the sentences, right? And that's because they are written in different languages and we are not versed with all the languages, but all of them say the same sentence. Same sentence is in different languages. It says we can do it. So imagine if the living beings on earth had, each of them was comprised of different letters or different alphabets. Understanding the entire ecosystem would have been so difficult, right? So that's what I want to allude to, that we are very lucky that all living beings on earth mostly are just coded by these four letters, A, G, C, T. You all know about it. I'll not get into the detail, but entire ecosystem. You look at animals, you look at plants, you look at birds, you look at bacteria. Everywhere the DNA is nearly the universal genetic material. RNA is there, of course, in viruses like the coronavirus of today, but nevertheless the rules are nearly similar. So there is a complementarity, and the advantage about that is, because everybody speaks the same language, if we are able to read those letters, then we will be able to make similar sense out of those letters, provided they have the same meaning, right? So just to recapitulate, all the cells in our body have all the 23 pairs of chromosomes, irrespective of which organs or which tissues are there. So clearly, if you take the genetic material out of any tissue, you will get in the human the same 23 pairs of chromosome. But then what makes one tissue different from the other? Clearly, the whole genome is there, but it does not everywhere express the entire thing. So some parts are expressed, some parts of the chromosomes are expressed in some tissue, some in another tissue, as a result of which you get a total kind of individuality. So this is what I was telling. Francis and Crick, you would have heard about, and you would have definitely, since you're teachers, you would know this. The structure of this genetic material was elucidated by Francis Crick, and that had actually been one of the major kind of contributions to the entire understanding of the genetic material. What is the molecular basis of inheritance, and it gave a lot of explanations. And a good part about it is that this is a universal language at the molecular level, and be it this, be it a mouse, be it any kind of plants or microorganisms, everywhere you have the same genetic material. So what is the advantage? Because they all have the same genetic material, you could use the same set of techniques to study the genetic material. And so if you are in a institute where there are genomics institute, you can pick up any plant, any animal, as long as you are able to get the DNA out of that, you'll be able to use the same set of equipments to study the entire genetic material, right? So um, so if I give an analogy as to what alphabets are to English, nucleic acid are to life. So you thread the nucleotides and you get this DNA, which you thread. Then you have genes, which have punctuations, right? And then each of these set of genes, which could be called as sentences, can be threaded together into a chapter, which are chromosomes. And if you put all the chapters together, then it becomes the one book. You get two such books from each one from each of your parents, and that entire thing is called the diploid genome. The haploid content is what is called as the genome, and the science of studying these letters are what is the science of genomics, right? So now there are interesting things, just like in book. If somebody writes a book again, mistakes will crop up. If it is written in some other language and then retranslated, then there we might lose some kind of essence. So this book of life, even though it's there and it's very ancient, it is there in the Africans also have the same book, Indians would also have the same book, but then there are subtle changes, right? So I would give an example that we start with a simple word called color, and it can have a deletion. Some places this can still have meaning, right? But then if you again start to have an insertion, then there's no meaning to this. And then if repeatedly it comes, then it makes meaningless sentences. So then there could be a change. The change could be one letter change, like a substitution, where you can get another one. Then you can also have other kinds of editing done, so that you can have a deletion. You can have another kind of substitution, which again might make sense. This can get inverted, and then sometimes it might also get fused. So what you start with some word, and then you get into another word like cool look, right? So this is what has happened to the book of life. It has it is it's there everywhere, but it undergoes changes within the species, between species, and that's what creates such a kind of diversity. Now, what are the consequences of these changes? So, as you know, this you would have, meaning you've been exposed to and you would have read. So, the DNA opens up to create a message, which is the RNA transcription. This process is called as transcription. And this is what is read by our machinery in the cell to make a protein, right? So not the entire genome is not made into protein. Less than 2% of the genome actually encodes or makes these kind of RNA, which can get translated into protein. So what happens? You have the DNA. This is written as AGCT. In the RNA, wherever T is there, it's replaced by U. So by base pairing, you actually make a transcript where A is there, U gets incorporated, where C is there, G gets incorporated. So like that, you make an mRNA, right? And this mRNA, three letters of these mRNAs are read something called codons, which can be recognized during the translational machinery by the tRNA, and it will get an amino acid and incorporate when it's reading these three letters. So like that, or there are 20 amino acids, and depending on what nucleotides are there, that amino acid gets incorporated. And when you thread all of these together, this is what makes a protein, right? So if you now understand that if that DNA has a small change, then that can make a different RNA. If that makes a different RNA, it will incorporate a different amino acid. And if it incorporates a different amino acid, the protein might change. And sometimes the change in the protein can be very drastic. For example, the beta globin gene. So there is a normal beta globin gene, and sometimes in this position 6, where in everybody nearly you have glutamate, if there is a change, it becomes valine, and there is a very drastic consequence of this change. So you have these RBCs which are spherical in shape, and they assume this kind of a shape, which compromises the oxygen carrying capability to quite a lot. So people who have these changes suffer from a disease called sickle cell anemia. So this is also uh, one kind of an adaptation also is there, because when you have this kind of a shape, then malarial parasites can get encapsulated there, and that's also a protective mechanism in populations which are endemic for malaria. So this is something where there is a change, but that change has also been utilized for something which protects you from malaria, right? But if you do not, if you have these sickling, then you also become anemic, and that's a major disease. It impacts a large part of the Indian population. So these are the few changes, or few of the things which people were noting, and they thought that if we can read the entire book of life, and we are able to read all the letters, which are around 3 into 10 power 9 base pairs, then we will be in a state to compare one genome with the other genome. And based on the changes, we might be able to say, okay, this person is diseased. If I go back to the genome, is there a change, and which can tell me what has happened because of which the person has become diseased, right? Or this person has a little bit of a different mannerism. Is it there because of the genetic makeup? And can I actually sequence the genome to be able to know that? So that was a major project which got initiated in the 1990s, which was called as the Human Genome Project. It nearly took 13 years. It was one of the, like the putting a man on the moon, that kind of a project for the biological sciences. And the cost of the project was nearly 2.7 billion to get the first draft of the human genome sequence. We have been able to read all the letters in the human genome. That was a mammoth project. It involved huge number of people across the world. And but even though it took 13 years, after that the technologies got so advanced that we've been able to now, if you go back to the go and look at the information after the human genome project, there would be nearly 500,000 human genome sequence available over there. What was very interesting was that even though we had so much sequence and we thought we got the prototype genome sequence, we found that eight genomes were actually mixed together to get a prototype genome sequence. And we found that even all the humans differ in 0.1%. But if you convert 0.1% to 3 into 10 power 9 base pairs, there's a huge amount of variation even within related members. So, for example, you have siblings, you might differ at 1 million places. So that posed another challenge, that now if I have so many changes, how do I understand what changes are meaningful and what changes are not? So now the science has progressed on that direction. But what was very important is that there were milestones. So human genome was not sequenced just like the human genome. So during the process of human genome sequencing, people had to learn how to sequence a genome. So lots of bacteria, yeast, small organisms also got sequenced. And there was a particular goal of the Human Genome Project that sequencing of the human genome, we might know all the genes which are encoded. So there was a mixed idea about how many genes code the human genome. Now it comes to something like 22,000 or something like that. Still, also the definition of genes is also changing, but we can take it that at this point, these are the number of genes we could consider on an average. There are 3 billion base pairs. So if we can read all the base pairs, and then we should have a way of storing it in the databases and to be able to use it for different kinds of application. And then this could be used for many different kinds of purposes. And I'll show you some of the applications, what people are using it for. And also there would be a lot of things to do with when do you know about the genome, how do you handle the ethical issues around there. People should not be using genome sequences to discriminate people, right? So there was a whole lot of things regarding ethical, legal, and social issues which might arise out of the project. So the Human Genome Project was not only to just sequence the base pairs of DNA, because if you sequence the base pairs of a DNA, you will just get in a computer AGCT, AGCT, AGCT written all over by 3 into 10 power 9 base pairs. And how do you know, or how do you make sense out of it, right? So that was not the only purpose of the Human Genome Project. The purpose of the Human Genome Project was also to understand. Now I'll give you some picture. Normally, again, I have this as interactive. Maybe somebody can write on the chat, also quick chat. Can anybody tell me what is this picture? Which place can anybody guess this picture is from? Anybody in the audience? Uh, okay. So I will maybe tell what normally people tell when I show this picture. They say that it's a desert because they see some
Rajasthan,
camels.
Bhuj, Delhi. This is what
Yeah.
Rajasthan, Bhuj, Delhi. This is the
Okay.
answer if you wanted.
Rajasthan, Bhuj, Delhi. Yeah. Because why do you say Rajasthan? Because you see camel. Bhuj also maybe because of that. Delhi, I do not know how people could make a guess of Delhi, but maybe the person has been residing in Delhi for quite some time, so they got some landmark. But anybody from any other part of the world can take it for some kind of a desert region, some kind of a rural region in Rajasthan where such animals are predominant, right? Now, if I kind of layer this information, this place with some more information, and I put this picture that this is nearly in the same place, will people be able to make out? Any guesses, sir? What people have written?
Yeah, Arabian desert. This is what he said.
What?
Pushkar.
Pushkar. Okay. So people who know it is Bhuj, they say it is Pushkar because you see marketplace, you see loudspeaker, and generally looking at the turban and all, you will see, right? I give you another one. Can people make out what this is? Now many people should be able to make out that this is what? Chandni Chowk, right? And now nobody can mistake. So that picture which I showed was Chandni Chowk of 1900s or something like that, and this is Chandni Chowk of today, right? So why I showed this picture is that when you sequence a genome, it's like Chandni Chowk. You cannot make out which chromosome from where, which part codes for what have you done over the period of time. You have layered it with more information about landmarks around that, right? As the landmarks got created closer and closer, you got to know, okay, if this is this near this, then that means it is in Delhi, right? So slowly, you actually populated the AGCT with certain kinds of places with some landmarks. And that's how, when the human genome would have got sequenced, people would know how to assemble the genome. So what comes from chromosome 1, what comes from chromosome 2, which is the gene, which is not the gene. So it is not that if you just sequence the whole genome into AGCT, you can make anything out of it. You cannot make anything out of it. So what you need some kind of things which will allow you to have landmarks, right? So that's where there is a portion in your genome, in your book, which talks about satellite DNA, which talks about microsatellite, talks about mini satellite. Those are the ones which were really key to the Human Genome Project. So if you take a DNA of human and you try to run it, earlier times when we used to purify DNA, we will run it through cesium chloride, Ethereum bromide, density gradient centrifugation. So what would happen is cesium chloride will form a density, and depending on where the density of DNA is, it will get layered around there, and then you use a syringe, and because Ethereum bromide would be there, you'll be able to make up which is where the DNA is. But then they found that there was another one which also was there in the DNA, but which separated out. Is my thing shows? Am I audible?
Yes, yes, fine.
Okay. So so what they found was that there was this hump, there was this another band, and which they called it as a satellite band. So what differentiated them is that the density of that region was a little different from the main, and that's because it had stretches of repetitive sequences which actually gave one kind of a density, because it was biased towards AT rich sequences or GC rich sequences. So there they could find that when they isolated that region out of the cesium chloride, the separate satellite, and then they characterized it, that they found that that was very rich in repetitive sequences of different kinds. So they called it as first classified it as two kinds of satellites. One of them they call as microsatellites, and the other called as mini satellites. So in microsatellite, typically you had a small number of base pairs repeated over and over again, 1 to 9 base pairs. So for example, CA, it is dinucleotide CA, 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 if it's repeated, right, 5 to 50 times, then it'll be called as microsatellite. But if it was a large repeat like AGCTGAGAC, let us say, and then again AGCTGAC, whatever. So if it gets repeated and it is a larger unit, then that would be called as mini satellites. Okay, so the genome was full of these microsatellites, and you can do a simple calculation. What is the probability of finding a CA in the genome, right? How frequently you will find, given that there are only 4 kinds of nucleotides, you have 3 into 10 power 9 base pairs. So what would be the probability of finding a C and an A together? It will be 4 into 4. So 1 in 16 base pairs, you'll encounter a CA. Then you add another CA, another CA. So like that. So there would be some places in the genome where you can get these repeats, right? So these repeats are there, and if they are flanked by unique sequences in the genome, then that becomes a repeat of a particular place. Okay, so this repeat, which could also, because it is repetitive sequence, when the DNA polymerase is replicating, it will make some errors, as a result of which the repeat sizes can change between people. So they started being when they characterized it, they found that the repeat length also changes, and between people, so they were called as polymorphic repeats. And when they had these variability in the repeats, then they were called as variable number tandem repeats. Okay, so what became that you have a genome in which you have repeats in a particular place flanked by a certain sequence, and if that repeat changes in size, then with the same flanking sequence, you will have a fragment of different length because the repeat has changed in number, right? So these are what was called as markers, right? So these were the ones, these were the very important reagents when people sequence the human genome, that what fragment of DNA comes from where, they could actually use these landmarks to create the entire, recreate the entire landmark of the human genome. So they started with making landmarks, they used techniques to sequence them. So I will just come to that. The other kind of landmarks were what are the regions in the genome which are expressing. So these are called as express sequence tags, because if there is a DNA and if that kind of transcribes into a RNA, then you will get an RNA sequence from there, right? So now supposing you take all the tissues of a person from cadaver or whatever different kinds of material they use, and they looked at the RNA content, and they had methods like reverse transcription, you would have read, to convert it into a cDNA, and you sequence it, then you will get a sequence from the expressing regions of the genome, right? So like that, you had small sequences coming up from different cell lines, and that formed a cluster of or a library of sequences which were called as express sequence tags. So what is the utility of that? So supposing you sequence a human genome in a particular way, and then you want to know in this human genome where this express sequence tag comes from, where you could use that as a probe and fish that out. So you can use different kinds of molecular biology techniques to be able to map the express regions using these tags. So not only these reagents were created as a resource for human genome sequencing, later these very same resources actually became very important when the assembly of the human genome was done. So typically what was done, that different cells, you extract the RNA, you reverse transcribe. So the mRNA is single stranded, you can make it double stranded by an enzyme called reverse transcriptase. And then you actually have it cloned in a vector, and then you can use the vector sequence to sequence it using a method called Sanger sequencing. I'm sure you have been taught that or you know about it. And then using the Sanger sequencing, you read these sequences. So these become a library of sequences which tells you that this is the expressed region of the genome, right? There's another method that expressed region of the genome comprises only 2%. What happens to the remaining 98%? So there was another method which was used, that you take the DNA and randomly fragment into small regions. You could fragment into different sizes, like the size of a yeast artificial chromosome, bacterial artificial chromosome, or a bacterial plasmid. You could clone larger fragment, smaller fragment, even smaller fragment, clone it into different kinds of vector, and then you sequence those fragments. So as a result of which, this was called as a shotgun method. You do not know where your fragments are coming from. You randomly make all the clones. You keep on randomly sequencing the genome. You get bits and pieces of your genome sequence. You find or you read those sequences, and then you have a broader map created by different kinds of techniques like RFLP technique or by marker, and then you stitch these sequences. So when you sequence these ones, they would have overlapping fragments because it is randomly cut. The genome is randomly cut, and when you assemble the overlapping fragment, you will be able to assemble the genome into bigger parts, the sequences, and those bigger parts then can be assembled into bigger libraries or bigger chromosomes. Okay. So this was roughly the way all the human genome sequencing was done till the Sanger method of sequencing during the Human Genome Project. And there were different kinds of landmarks. So Human Genome Project was not done in a day. It was done in 13 years. And that is because at every point of time, considerations have to be made as to how you, so it was launched in 1990. Lot of deliberation, what to sequence, whom to sequence, where to sequence, what technologies to use, how to increase the throughput. So all those developments happened over a period of 1995. And then they said, okay, let us do a pilot study. So let us sequence the Haemophilus influenza, the first bacterial genome, or sequence yeast, which is a eukaryotic one, or sequence mouse. So that way, people started participating, and people started sequencing different ones. Then they sequenced the first chromosome. So the first chromosome was the smallest chromosome, which is the chromosome 21 and then 22. Both of them got sequenced. So they learned how to sequence and assemble a full chromosome. And by the time 2001, the first draft of the entire genome was there. So from the first human to the first draft, this was the final phase of the Human Genome Project, but it took nearly 13 years to reach that place. And as you can realize, because you needed too many things. You needed a technology which will sequence at a pace, because if you have to read 3 into 10 power 9 base pairs, when they started the sequencing, the technology was all radioactive based. So there was a fluorescence based method were invented, and then that could increase the number of sequences you can read at a particular time. Then there were these big automated machines, like this kind of instrument, which could actually deal with lots of sequences at a particular day. And then there were huge number of labs all across the world. They participated to complete the sequence, but that was the first draft. After that, it required lots of finishing, and then the final version of the human genome sequence came out in 2003. So this was the total human genome project. But then what happens? Meaning, did they meet all the goals which they started with? So they got to know as the sequencing progressed, that as they were predicting, less than 2% of the genome codes for protein. But then in the process, they actually identified that there is too much variability in the human genome. And what is also very interesting was that the human genome was full of lots of repetitive sequences. These repetitive sequences were not only the simple repeats, which we talked, but lots of transposons, lots of retrotransposons, and these simple interspersed repeats like the sign and the line. So these are like small genes repeated many times over in the genome, and they code independently. So those kinds of and so you can very well see that the human genome pretty much has so much amount of other kinds of material, and the coding part, as we understand, is the protein coding comprises only so much. So so then then people thought that if they sequence the human genome, they will know why they are superior, because there were a lot of paradox during that time, that why we are so superior. Is it because we have more number of chromosomes? The answer is no, because even a grass has 12,006 meaning 1260 kinds of 12 these many number of chromosomes. So clearly the number of chromosomes does not contribute to complexity. Then people thought, okay, humans might have more genome, because of which they are more complex, but that also got demolished, because amoeba has more, and you know what amoeba is to human, right? We think so. So then people thought, okay, when the human genome gets sequenced, we will get to know that humans have more number of genes, because of which we are complex. And then it turned out that also was not so. Like Arabidopsis or Drosophila, Arabidopsis in human, they roughly have the same number of genes. So what was different? So it turns out that repetitive content is something which is increased as the humans have become complex, and that we can have it at a later point of discussion. What do the repeats do? But those are the places now people have started looking for, that what makes a person human. How do you define a human genome or human per se, right, from the genome sequence? So now I was mentioning to you about the markers. So the same markers which were used for human genome project to be able to know the anchor points also became useful after the human genome project came out. So some of the usefulness, I think you have in your textbooks of DNA fingerprinting. And typically, earlier times when people did not have the sequence, they used to use restriction enzymes, which would cut the genome into different sizes. And based on the differences in size, you could tell that, okay, this person has lost another restriction enzyme site. This site is lost. So there is a bigger fragment generated in a person. Whereas if a person is normal, then if you use the same restriction enzyme, it will cut again in the middle, so you will release a fragment, right? So this was so for this you needed to have lots of restriction enzymes and lots of ways to cut the genome and to be able to know that. But now as the human genome sequence has come up, you could actually sequence bits and pieces around the genes of interest, and you could read the sequence and you could say whether there is a change, right? So that's the change which has happened when the human genome sequence has come out, because now you know the entire genome. And then if you are able to map certain regions which are different in the disease versus normal people, then you could sequence that part and you can really go and look at what is the change there. You could use these things for also paternity testing. One of the things is the variable number tandem repeats, because they vary in their size between people. So if you actually do a PCR amplification around different variable number tandem repeats, using the unique flanking sequences, you actually get a fingerprint of the person, right? And that fingerprint will be different from the other person because of the size of the repeat. Okay, some might be missing, or some the sizes might be different. Now, if they have progeny and you want to know whether those are related, then one should come from the mother and one should come from the father. So if all the bands in a child gets explained by their presence from either one of the parent, then you can say that this person is most likely related to them, right? Whereas if you have different kinds of bands coming up which are not present in either of the parents, then you are able to know that this is different. So this has utility in paternity testing, and this has also utility in forensic. For example, in a crime scene, if there are samples available from, for example, hair or semen or whatever it is, you can isolate pretty much DNA out of there, and then use the same kind of VNTR sequences to amplify. And then you can see whether at the scene of the crime, whether you are getting another DNA, so you know definitely that there is another person around there. And then if you have prospective kind of criminal known, then you can take their DNA and you can match them, and you can see which of the person's DNA matches, and then you might be able to recreate the crime scene, or help in elucidating those cases, right? So this kind of a so now the human genome sequence got sequenced in such a kind of machine, but now there has been lots of advancement, as I told you, because that was 2001, and now the instrument sizes have also become very small. So this was the first generation sequencing, then there was a second generation sequencing, then now there are these third generation sequencing. It is becoming more and more miniature. You can read many more sequences at a particular time. So what took 13 years in these kinds of machine, now we can sequence 24 people in one day or 2 days using these kinds of machine. These are the size of a spectacle case. So we have this in all these in our institute, and when the lockdown gets over, anybody is interested, you can actually, we have a museum also of the old equipments, and we also have outreach programs where we take you through all these things. So what happened during these years? So not only human genome, lots of eukaryotes, prokaryotes, pretty much everything from the whole tree of life has started to getting sequenced. And each of them sequences, people are also getting to know what are the different kinds of stories. Like I started with that, they found what is component of the royal jelly, which genes are involved in forager bees, what kind of genes. So there are genes which are increases the vasculature, which actually leads them to accumulate so much water. Or there's this bacteria which can withstand radioactivity. So the way they found is that it has a very efficient genome repair system, as a result of which if there is a intense radiation, it can repair its genome, and that's why it can thrive in very adverse environment. This kind of an information is very important, because then you know which are the genes which are important in DNA damage repair, and some amount of applications one can think through that. Or what is the size of a very small, a minimal genome. So there's this Utricullaria gibba, which feeds on insects, and it has a very small genome. It has got rid of pretty much anything which it doesn't need, and the genome size is very small. So each of these genomes, when they are getting sequences, newer and newer stories are coming up. What made human bipedal, right? What contributes to the larger brain size? What are the kinds of surprises humans have? And if humans have so much of repeat, then compared to the other one, those repeats, do they have function? So once you have these, lots of questions can be asked. And lots of other kinds of interesting things are also coming up. For example, there are animals which have pigmentation defects. If you know the genome of that, then you can also look at the human diseases which have pigmentation abnormalities. Do they actually share the same genes, right? So there is a newer area of genomics called as phylogenomics coming up, where people are using elephant genome to understand why they don't have cancer, what is it in them, can you have then better therapies if you have that understanding, or many kinds of conditions. Okay, I'm just trying to give you a flavor of the genomic area. So for example, there is this new initiative which is called as Earth Biogenome Project, which all over the world plans to sequence 1.5 million eukaryotic species on the earth. Or there is this Darwin Tree of Life project in UK, which actually wants to sequence all the species because they want to know how ecosystems come about, right? Why something is dwindling. Is it that it is tied up to loss of any other thing? So that kind. Or Chinese and other initiatives have started where they sequence 10,000 bird genomes. You get to know different birds have different plumages, eating habits, feeding habits, etc. So each of the genomes are telling each story. Then I told you about whether you know the past. So there's this person called Stefan Schuster. He does something very interesting. He calls that area of research as museomics. What he does is he looks at the sequences of endangered animals by taking museum specimens from where DNA can be isolated, and looks at them all across the world and tries to see whether animals, when they became extinct, did they have something in their genome telling them that something is becoming extinct. So he worked in this Tasmanian devil, which have just got extinct in the last few years. And then he could find that in the mitochondria part of the genome, there are certain signatures that it becomes very homozygous when it is on the verge of extinction. So using this information, he actually could work with some conservationist in California and could increase the number of these birds, which had dwindled to 40 to 400 by captive breeding. So learning lessons from ancient DNA is actually providing solutions to newer and newer things. Then forensic, I told you already about. There is this very interesting story I just wanted to relate. This is like closing 500 years plus missing persons case. How genetics is actually being used to identify or solve some cases. And this was of Richard the Third, who died, but people did not know where his coffin was, and they discovered a coffin a couple of years back in underneath a garage, and they thought that all the bones, archaeological radioactive dating suggested that this is of Richard the Third. Then what they did was they actually looked at a 19 generation removed female and a male, and they could actually from that lineage trace that the mitochondrial DNA was shared. So that was one level of evidence. Okay, it could be Richard the Third's coffin. And then they looking at the genome, they could also predict what eye color or what hair color could that be, and that there was a portrait of Richard the Third. They could match it with that. So putting all these informations together, because the DNA was available, living person, very many generation removed was there, they could actually tell that that was Richard the Third's coffin only, and that actually put a nail to the coffin, literally, that they got to know that this is Richard the Third's thing. There is other kinds of things which the Human Genome Project has come about, told us that we used to think that whatever cells are there in the human, they only comprise human. But now it turns out that we are colonized by bacteria in all over our body, and they are in different places, but the same kind of bacteria colonize different people across the population. So if you're human, you have such kind of bacteria colonizing such places. So now, because of genome sequencing, you can actually sequence a colony of bacteria from particular niche and try to find out what colonies are there. And it turns out when you're 8-month-old, the bacteria starts colonizing. And for a person, the bacteria becomes a signature. So if a person is thin or if a person is fat, people have found that there are differences with respect to microbes and the microbial genome, which makes them different. So this is the area of metagenomics, and this is something which is very interesting, because how they know about this bacteria is that all the bacteria have this 16S rRNA sequence, and the 16S ribosomal RNA is different amongst different bacteria. So if you only sequence that 16S rRNA, you will be able to say which bacteria it came from. So you don't have to culture the bacteria, just by looking at the sequence, you will be able to know where it comes from. So now when people knew that this is there, then they thought of a very clever thing, that they found that leeches suck on the blood of many different animals. So if you took the blood out of the leech's gut and sequence that, then you will be able to know which kind of animals are there. And like that, people have been using the same approach to find out from the animals which are there in the forest whether they can count which kind of animals are there, because the mice will, or these will feed on that. And when you know the DNA, you can match it to the whole library of DNA sequences and get to know what kind of animals are there. So people are using this for tracking animals. And there's this very fantastic story of how you get to know when people started wearing clothes. So it turns out that in our we have lice in our body and lice in our hair, and lice obligatorily feed on fiber. Okay, so there was this scientist in Germany who had a very crazy idea, I'll say, that he thought of, that if you sequence the lice from head and from the body, then you will get to know when did the body lice arise, right? Because if body lice has arisen, then surely there would have to be a fiber in the skin to be able for them to be able to feed, right? So looking at the mitochondrial genome of head lice and body lice, they were able to actually find that the body lice has arisen maybe 40,000 years back. And then they got to archaeology evidence where needle was there, and they could actually say that maybe people started wearing clothes 40,000 years back, because that is the time the body lice also came. So now you realize how the knowledge of all these animals around us also tells some very interesting stories about humans. So I decided to keep this slide over here just to tell that people are using genome sequences in many different ways. The latest one is coronavirus. We've been sequencing coronavirus genomes to be able to know what kind of viruses came to our country, whether viruses which are there in our country are different from viruses outside. In different parts of the country, our virus is different. Because if we know the sequence, then we know the regions which are kind of needed to be able to amplify and to be able to detect and do all these contact tracing by RT-PCR method. So in India also, lots of initiative has happened. Nearly 1,000 different kinds of coronavirus genome from different patients have been sequenced. And for all of you who are interested in seeing this looks like, if you are in an aeroplane, you will find this is how they show the movements of their flights, but this is actually coronavirus genomes from different parts of the world. And the based on sequence similarity, they are able to trace if something originated in China, where did it go? From there, if there was a change, where did it go? So you can actually go to this site. It's a very interactive site, and you can actually follow or track how different coronavirus genomes have come across in different parts of the country. There are these phylogenetic lineages, so you can visualize it in different manner. I just thought that because it's a coronavirus time, I should kind of try to show you there are different resources available, and the kids might really connect to it if you're able to tell a very topical thing. So this is how typically a tree looks like, that which is original one, after which what diverged, and then what diverged, and these are different parts of the world, what kind of mutation arose. So this is a very interesting, and this tells a timeline from December to March, how each of these lineages have arisen. So you click on each of these dots and it will tell you which individual from which part of the country has that sequence has gone to contribute to this kind of an understanding. Now, a little bit about differences. So that was about genomes. You have genomes, you can annotate, you can use it, but then you have differences in the genome, one individual to the other, between human and chimpanzee. So you need to know what differences makes us human, what differences makes us different. For example, if you look at this is a map of solar radiation. So this is more intense solar radiation, and as you go up, there is lesser solar radiation. And if you look at this map is like the solar radiation, but what happens is that this is actually the color of skin. So this is an adaptation to be able to protect yourself from UV. So places which are near the tropical region, people have dark color pigment. Now, if you go and look at the genetic makeup of these people, then you discover certain genes which has some changes between the Europeans and the Indians and the Africans in this kind of, this is a gene called SLC24A5. The name is unimportant, but if you have a single change in this gene, this actually differs between the Africans and the Europeans. And people have done studies with this kind of a genetic makeup, and they found that this is linked to lighter color pigmentation. But what is very interesting is that the same gene variation is actually linked to differences in pigmentation in all these animals. So like I said, if you know if it, because it's a universal letter, and if the universal letter forms a universal sentence, then you actually can be able to ascribe genes which are linked to skin pigmentation. These are very important in diseases where you have loss of skin pigmentation, etc. So this is what the genomics is leading about how from adaptation you can link to genes, or you can link to how people have moved. So this is archaeological evidence that we know 130,000 years back, people were there in Africa. Slowly they moved. So when they moved, like I showed, there are changes in the genome. So now if we take genetic material from different parts of the world, can you actually see how distant they are from each other, right? So this is a genetic map of people from different parts of the world, the Africa, Middle East, Europe. So each of these, there's a line of, every line is one individual. So if the individual has the same color, that means they are more related to each other. And in another part, if the color is changed, then that means the genetic makeup has changed a little bit. And then another change, so it shows that there is a little bit of this European into Middle East. So they are admixed, and these people are nearly homogeneous. And then there are South Central Asia. So as the humans have moved, the genomes have changed, and those changes can also be seen as genetic signatures.
Okay.
Why this is important?
I am attending Zoom.
Hello, hello.
Oh no, please carry on.
Okay,
Carry on.
okay. So if there is a change, then that change also moves along with people's movement. So once you know which populations are more related to each other, you can do many more things with it. So this was a world map. So in India, we also created a map of Indian people, because India also, there's been different waves of migration, different civilizations have existed, different time periods. So we did not know, if you say what is the genome of an Indian, is this one genome or there are many genome? How diverse is the Indian genome? Is it as diverse as the diversity of the people? So this was what we did. This was called as the Indian Genome Variation Consortia project. I was the convener of the project, so I worked in this project for quite intensively, and we could make actually a map of Indian genome. So this map is very interesting. The down below this is the linguistic map, that all the people in the blue region speak Indo-European language. These are the people who speak Dravidian language. Like that, we have a linguistic map. And then these all other populations or places where we collected samples from 55 countries we studied, from different language, different ethnic background. And we asked the question, are people who speak the same language, are they more related? People who reside in the same region, are they more related? And then you can see that here the colors are all over the place, right? So a pie chart, if it has the same color, that means they are more related with each other. So here you see people related with each other, they are all Indo-European. But here also you see another color coming up, that means these people are different from these at the genetic level. Okay. And then you see in the same high altitude, there are other sets of people who are of a different color. So that says that even within the northern region, if you are Tibeto-Burman or you come from Kashmir or you are from this part, there is a genetic difference. But these part people, some are also related to these. So we went back to history and we found that there had been invasion. So Dravidians were either pushed up or pushed down. So there are some populations which Kashmiris and some Dravidian population actually have similarity at the genetic level, but these are older times, right? And then these are tribal populations in the middle part of the country, which are more similar to each other. So like that, we created a genetic map. Now, how does that map help us? So I'll give you just maybe one example here. So this is a map of malaria endemicity. This place has got very endemic for malaria, less endemic, and this is not malaria endemic. Now, this is a map which looks like malaria endemic map, except that this is a map of a genetic variation. Now you see there is intense color here and not so intense color here. What this tells us is that if people are residing here and they have difference with respect to this genetic change, then it might be protecting them from malaria. So if you have this information, then you can find what protects them and whether if people are susceptible in other parts of the country, can we use this information to screen who are the people who are susceptible or have diseases. So like that, we made different kinds of maps for protection against HIV or diabetes susceptibility gene. So like this, once you have a baseline map, you can actually do lots of interesting things. And this is where why people are interested, because if you know about these variations, then you can know who are the people who would be qualified as normal. If there are changes, will they be in the pre-disease state? If there are more changes in the disease onset, then can you predict who are the people who are likely to have a disease? How will they progress to disease? So can your genetic makeup tell you all these kinds of things? And can you use this information to prevent diseases, screen diseases, predict how the disease is going to happen? How do you monitor or how do you customize life? So basically, how you maintain health or how you maintain quality of life. So this is what is called as precision medicine, that if we know our genetic makeup, then can we actually look at the changes and are able to predict from the genetic makeup and tailor a person's or customize a person's disease. So this is something which is given in Ayurveda. Today I'll not talk about it, but uh, this kind of a principle of keeping people healthy and all, these are shloka from Charak. And what in our institute for the last 20 years we've been asking, what is the basis of this precision medicine in Ayurveda, and can we actually use genomics to integrate these two sciences together and try to understand how Ayurveda people treat people in a customized manner, and can we integrate it with the modern genomics. So this is a new area of research which we have initiated in the country. It's called as Ayur Genomics. There's a colleague of mine, Dr. Bhavna Prashar. She's an Ayurveda doctor, and with her, I have developed this area for the last 20 years. Then something which the school kids would be interested, definitely, there's an area called precision nutrition coming up, which actually it's the same genetics, but now you look at the genetics in the taste receptors. So it turns out that even in a burger, if there are different components, we have taste receptors which sense these components. So if you have differences in these taste receptors, then your ability to perceive sugar or the glutamate in the tomato might differ between people. And that is why you might have want to customize your burger in a different manner. So people are using this kind of an information to know why people gravitate towards certain kinds of food and why they avoid certain kinds of food because of their taste receptors, and how having that knowledge actually governs what you eat, and what you eat governs what microbe you will have. And so if you knew about these changes, then you might be able to change your eating habits and maybe change your microbes, which have an intricate link to your mental state. So you can see entire ecosystem is correlated to each other, and many of these changes stem from what your genetic makeup is, right? So having a knowledge of the genetic makeup sometimes might also allow you to make better decisions in life. For example, there is a gene for bitter tasting. So if you have a very bitter taster, then some people like will taste of bitter, will avoid bitter food. And if they avoid bitter food, they will avoid all these kinds of good food. They might avoid alcohol, they might avoid smoking, but they might end up liking lots of sweet. So what happens is, because of this genetic change, you will have different eating habits, which might give you risk for BMI, metabolic risk, etc. So having these knowledge about changes, you might be able to have a better mindful eating also. I'll just come to the last part, that how this is helping in diseases. So this is one disease we've been working for the last 22 years. Disease is unimportant for the subject matter, but this I'm just telling you because we've been working in this disease. This is a neurodegenerative disorder which actually leads to movement problems, and then people become wheelchair bound. It's a progressive neurodegenerative disorder. It runs in the family, and it's a very sad kind of a disorder, because not only it runs in the family, subsequent generation gets the disease much earlier. So a parent might have retired and he might have a child who has this problem, and not knowingly, because if the tests are not available, you will not be able to know that you carry a mutation, because you will not exhibit the symptoms with a lesser number of typically this disease happens not because of one change, but that change is because of a repeat sequence which expands during transmission. So this person would have at a particular place a repeat which is of 30 size. In the next generation, so this repeat is something like a mini satellite, but this is in the coding region. So it increases in length, and it increases in length, then it presents with certain symptoms. In the next generation, the length again increases. So like that, over generation, the length keep on increasing. And as the length keep on increasing, the disease becomes much more severe and much more earlier. So we've been working in this kind of disease. We've looked at nearly 5,000 people across the country in collaboration with AIMS. And we've been studying all these big families where the disease runs in the family. And then we try to see who are the people who have got the disease and whether they are likely to transmit it to the next generation, which is asymptomatic. That is, they don't have the disease as yet, but since it is progressive, they are likely to have the disease. So we've been using this kind of a genetic method or genome sequences to identify such kind of genes which runs in the family and causes such kind of disease. Because once you know the genetic basis of disease, then you can actually have a help to a community where because of marriages within the community, one kind of mutation got increased in a large number of people in the family, as a result of which many families are suffering from such kind of disease. So this is a family in Buldana, 200 families in Buldana we identified who are suffering from the same disease, and they did not know, they did not have a test available, because of which it kept on spreading in the families. And now we've been able to at least identify, and then we are trying to go back to the community and trying to see how we could help them. Last exciting part of the information, that the DNA, as you can understand now, very small amount get encapsulated in a cell. So people are thinking whether if we run out of all these solutions of storage, can we actually think about DNA as a information storage material? Because in a very small tube, because the sequencing cost is reducing. So if you actually synthesize information, put it in a tube, keep the tube in the fridge or whatever, take it out, sequence it, and uncover the information. Like that, libraries can get converted to DNA libraries. So to test this out, this was a very crazy experiment. This was published this year, and I thought I'll end my talk with this. So people thought, is that doable? Can you actually store the information? So what they did was, they wrote a code. So you know this 3D printer, right? 3D printer needs a command. So what they did was, they took the command in the 3D printer and converted it into a DNA. So all the languages can be converted into this binary code, right? the binary code can be converted to DNA. So the computer language can now be converted into a DNA language. So what you do is the command to print a rabbit in the 3D printer, you convert it into a DNA, and you synthesize this DNA, and you encapsulate this DNA into a nanoparticle, which goes into a material which is used for DNA printing. Okay, and then you shoot the command and you make a rabbit which has a material with a nanoparticle encapsulated in them containing information about how to print a rabbit. Okay, then you take that plastic out of it, take out the nanoparticle, take out the DNA, and read that in the sequencer, convert it into binary code, and again give a command and try to see whether you are able to replicate again rabbits. So this is like a fiction, but this has been achieved. It has been done in Stanford, and this is what they call as DNA of tomorrow. Instead of Internet of Things, it is DNA of Things. So this is what I wanted to leave you with, that if you understand the genome, then you can understand lots of things from the environment, niches, how it adapts, how admixture happens, how the diversity happens, and how that diversity creates patterns, which can be telling you about health, about phenotypes, about disease. Not only that, you get to know about history, ancestry, culture, climate change, ecosystem dynamics, all these things, and maybe tomorrow's digital data storage solutions. So thank you. I'll end here and happy to take any questions. Thanks.
Thank you, Mitali. Excellent presentation. I have been getting messages in the chat box. Wonderful. And there are a couple of questions.
Sure.
But before we take questions, I have two announcements to make to the participants. We are planning to give you certificate for participation. But for that, you will have to send name and address by email to the Genetic Trust. If you can send that, then we will send you the participation certificate. And also for the Saturday, we have interactive session where the Dr. A.K. Singh, the Director of IARI and the Director General of ICAR, Dr. Mahapatra will be participating. We want 3 participants to express their views. Please give names of 3 participants who will express their view about this webinar as a feedback on Saturday. Discuss among yourself and communicate to us. These are the 2 announcements I had to make to the participants. Now we will take up the question answer. I have tried to pick up the questions. I'll read one by one and you can answer. One of the questions is, what's the significance of 98% of genome does not code for anything, including the introns? So what is their significance?
So as I was telling in the beginning of the lecture, that there are dogmas, and those dogmas got established because very prominent people talked about those dogmas. So Watson and Crick had negated that repetitive sequences are junk DNA, and from that time onwards, everybody started thinking that those are junk DNA, so they have to be neglected. But over the last 20 years, people are finding more and more functions out of those long non-coding RNA. And there is a whole lot of new functions coming up. In fact, very recently as last year, people have thought there are these dead genes which are called as pseudogenes. They have no function. But if you knock out a pseudogene in a mice, then it becomes lethal for the mice. So it is not that they are not coding. At this point of time, we do not have adequate information to say how much is coding, how much is not coding. And lots of long non-coding RNA, their levels get elevated in different kinds of infection, different kinds of times in temporal conditions during infections. That is not true anymore, that the remaining 98%. So in fact, after the Human Genome Project, there was another project which was called as the ENCODE project. which looked at, let us look at all the functional part of the human genome, and they came up with that the 98% of the genome is functional. So all the long non-coding RNA make an RNA, and the RNA makes a product which could be either as an RNA it can function, or as an antisense RNA it can function. So there's a lot of function coming up. That can be a different lecture totally. I work on one such repetitive sequences called allele repeats in the human genome, and we made some exciting discoveries there. So,
Yep.
so happy to speak later about that.
Yes, yes. No, no. In fact, the junk DNA seems to be a appropriate term again, because junk is not a waste.
Yeah.
Junk can be revisited to rescue
So
something.
it's now called as scrap.
So that maybe a spoon.
Yeah.
Yes. No, you can take parts and use it somewhere.
Yes.
So this might be an appropriate terminology, because
Yes.
it has significance at some stage of life or some stage of the evolution.
Yeah. So there are wonderful reviews coming up. How we should be looking at junk very respectfully. So
Yes.
there are very beautiful articles on the net about junk DNA.
Yeah. The other one is that even ataxia and other this one. Often we say that error occurs during replication. So why this error occurs, how often it occurs, and in the sense, people are not able to get to the idea that over generation it increases. That means every time it happens. So give some idea as to the frequency.
So the repetitive sequences are so the ataxia is a particular different kind of a disease. Not all diseases are caused by repeat sequences. Actually, majority of the Mendelian kind of so this follows a non-Mendelian inheritance. That means whatever you see in the parent, in the next generation you see it as more severe. Other kinds of phenotypes get impacted. So it is not really a Mendelian disorder. It's a little different disorder, and these are called as repeat related disorder. Here what happens is, because the repeats are repetitive in nature, AG or CAG, when the polymerase tries to read it, sometimes it slips. Okay, and we have excellent proofreading machinery, but if the proofreading machinery is not working very well, then errors start creeping in. So whichever cells divide for longer time, so if there are sperms, people have shown that younger children, if the repeat is expansion is there in the male members, then the younger children of that person has a larger length of repeats, because over a period of time many of the sperms will actually have larger repeat which will get transmitted. So this repeat happens across generation. In certain cases it also happens in tissues. So there is a disease called myotonic dystrophy. It causes muscle weakness, and that is also caused by another repeat expansion, but the repeat expansion becomes much more unstable in the muscle tissues. So there are these germinal ones also, there are somatic ones also. So it will depend on how good is your proofreading machinery and which context the repeats are, mutations are, you will accumulate that kind of a disease, or that kind of a change.
The next question is, in the course of your lecture, you just mentioned a sentence and somebody has picked it up. Gene definition is changing.
Yeah.
We want to know that.
So gene definition has changed over a period of time. Earlier it was one gene, one protein. Now it is like any segment of DNA which can code for a function. It could be protein, it could be RNA, it will be defined as a gene. So what traditional gene definition of that, it codes for a product, right? But if any DNA fragment is functional, then that kind of a definition is also getting defined as a gene. So traditionally, but when people ask you in your exams, then you'll have to stick up to the textbook definition. But then if you go and look it up, even I am saying something today, it would also change tomorrow. It's a very dynamic area. So this is just to prepare yourself that if you are teaching such kind of a subject, it is very essential to go back to web and look at the literature when you are teaching the students, because it's a very dynamic area. And I think if I write a textbook in this area, I really don't want to write a textbook because I have to write a textbook every year because the whole information is changing. How you did sequencing, today's generation student, if you ask about sequencing, they have not seen those instruments which we use for sequencer in our PhD time. So it's
Yes. I would add a sentence of caution to the teachers, particularly. You please do read, as Dr. Vitali has been telling, but don't tell everything to the students.
yeah,
So
yeah.
you will get
They,
confused. You need to write answers as given in the book. Otherwise, they might lose marks. So please do read for your information, but not for transmitting everything to the students. That judgment has to be made. It's good to know, but not necessary to explain everything to the youngster. A couple of simple questions. What's a pseudogene?
What's a pseudogene? Pseudogene is considered to be a non-functional dead gene. So typically earlier, many genes have different many copies, and typically if that codes for a protein, you translate that other copy, it would encounter a stop codon. So people thought so that was given a definition as a pseudogene, that it is same as a gene, but it is not a gene because it would not code for a protein product.
Okay.
Now, people are finding that even if it doesn't code for a protein, it will transcribe as an RNA. And if it transcribes as an RNA, sometimes it becomes a competitor of the main gene because it will kind of sponge the important proteins required for the main gene. But the traditional definition of a pseudogene is something which is false, which is not that kind.
Okay. Uh, how much variation is found between two individuals? And can we have a human kundali?
Can we have a
Human kundali.
human kundli?
Kundali.
Kundli. Okay. So even between siblings, you might differ in 1 million places. And total, when people have sequenced so many genomes across only the single nucleotide polymorphism, so they are chromosome duplication, inversion, many copy number variation, different kinds of changes are there. If you take SNP variation also and you aggregate all the SNP variation, common variation across the world, there are 38 million or more. My numbers, that number also keeps on changing as more and more genomes get done, right? And as regard whether there is a signature of a person, yes, if you actually sequence enough number of VNTR and enough number of SNP, there is a fingerprint of every person. But whether that fingerprint is predictive of certain diseases or certain health states, which is what you might say about a kundali or whatever, you can forecast the future. So certain kinds of places, when people are doing enough number of functions, having a change can forecast whether the predisposition is likely to be different. But that also has to have a cautionary note that there are sometimes many changes which also come with compensatory changes. So unless you have studied it enough, you will be maybe sometimes make false predictions. But mostly if you are studying rare diseases where one mutation explains a disease, then you can, if you know about that variation, then you can predict in the next generation if the person is likely to get the disease. But now people are again finding that there are modifier changes sometimes, because if these changes are very deleterious, then there are compensatory changes. Then it is possible that the person might have the change but might not present symptoms. So when 1,000 genome sequencing has happened, people have found that on an average 100 to 105 very deleterious changes are there in normal healthy individuals also. But in the context of a family, it might become a rare disease.
Uh, can you just recapitulate DNA storage? Somebody wanted
DNA storage. So
DNA.
DNA storage is nothing but, so in a cell which is very compact and the nucleus is very compact, that houses 6 into 10 to the power 9 nucleotide sequences, right? So that's a huge amount. Now if you, and that's from one cell. In a person, if you actually take 10 ml of blood, the amount of DNA you will get, that will occupy the only one small pint of or a drop in an Eppendorf tube, in a small tube, but it houses that much information. Now, 3 into 10 power 9 is a huge information. So if you convert your binary code into AGCT, then a big language or big sentence, you can compress it into a small, into a stretch, which can get converted into a nucleotide sequence. And if you were to clone that nucleotide sequence, you will be able to put that in a tube and it will occupy no space. So our entire book is not 3 into 10 power 9 letters most of the time, right? It's many books put together. So you can see how much information you can compress it into a genetic material. So what you could do is you can synthesize this DNA. Artificial DNA synthesis can happen. So you can actually create this information or encapsulate this information in a DNA string, and that string can be cloned into a bacterial plasmid and can be stored in a small tube. So that is what is called as DNA storage. So when you want to kind of retrieve that information, what you have to do is just take out that plasmid, amplify it, and sequence that small bit. And because you might have a code for what that sequence stands for, if once you read the DNA sequence, you can convert it into binary code and read. It can be either an image, whatever. So your entire image, pixelated images, all of them are binary codes at the back, right? So that is what is the DNA storage solution people are thinking about.
Okay. Two, questions which are not of really direct to this one, of a wider this one. One is how genomics is contributing to eugenics and euthenics.
So I would not get into that discussion.
That's what I said.
There are good changes, like for example, rare disease. If you are able to change the mutation and are able to have a person treated of sickle cell or beta thalassemia in that way by CRISPR-Cas, those are the advantageous ways you can use the
Yes.
genome sequence for kind of bringing to health improvement, right? There are other things like it's called epigenetics, where good diet can actually change the way your genes are expressing. And maybe if they are linked to reducing inflammation, increasing your immune system, those are the ways by which you should learn to modify the genomes, right? So that's the thing. I will leave it at. There could be many more
Yes.
things which can be done with the DNA, but uh, but that's also a good part of that part about NS, right? It's like the nuclear fission.
Yes, because genomics is a very important weapon also.
Yes.
So that's why everybody is now talking about caution as to where to go, where we should.
Yeah.
Lots of things are happening.
Yeah. So that's
We all
why
know about some genome editing
that's why I mentioned that one
in
important
China.
one important part about the Human Genome Project was the LC, the ethical, legal, social implications. So anytime we do genetic study, we have people diagnosed for a genetic disease. How do you reveal the test report? Whom do you reveal the test report? How do you communicate your research finding so as to not castigate a population? Because insurance companies can use that in different ways. So there is a whole lot of ethics beyond just doing the genome sequencing, right? And that needs to be really thought through.
Yes, I think we have taken up almost all the questions. Uh, I thank you once again for an outstanding presentation,
Thank you, sir.
a very comprehensive and interesting presentation, uh, because I can see lots of people sending their appreciation via chat. I'm just conveying it to you.
Okay.
And we hope we'll have many more sessions on various aspects. Uh, more as an informative one rather than education for the teaching purpose. As I said, one could be more enlightened, and that will make a difference in terms of teaching and also mentorship. With this, if you have any final words to say, I give it to you, or else I'll give it to Deepak, who is holding the controls. Any last words, Mithali, from your side?
Thank you, Mitali.
Thank you, sir.
Wonderful presentation. I thoroughly enjoyed it.
Thank you, sir. You are the teacher. So,
Okay, Deepak.
Okay, sir. Thank you, ma'am. We will end it, sir, here.
Okay, thank you, everybody.
okay. Thank you, sir. Thank you.